Today I'm going to head back heavily into life science and talk almost entirely about protein structure. Very little mathematics, if any. And it turns out that there are three big types of proteins. Fibrous proteins, globular proteins, and membrane proteins. And you might see that one of these three is missing here, and that's because I'm going to cover it in detail tomorrow. But before we head into that, let's spend the time we need on our normal discussion to see where we were yesterday and if there are things you want to discuss in more detail. I would almost argue that this is more important than the lectures, because most of the stuff in the lectures you can read from the books and lecture notes. And you're passing around the handouts. And I should have put numbers on these. Sorry, so you're just going to have to read them aloud and say which ones you're picking. Yep. Difference between thermodynamics and kinetics.
So thermodynamics considers states at equilibrium and kinetics not necessary.
Yes, and in particular, we discussed this in relation to the free energy landscapes yesterday and the free energies. So there was even something more you could say about this. That it's entirely correct what you're saying, but this means that there are different parts that are relevant for them.
Transition rates?
So transition rates enter where?
In kinetics.
And for that, what part of the energy landscape is relevant? If that is wrong, you get one more chance.
Oh, you mean...
Yeah. So the point when we're talking about kinetics, you're talking about transitions, and then we're talking about barriers. So surprisingly enough, with kinetics, the worst part of the free energy landscapes, the ones where we virtually never spend any time, they are almost the only ones that matter, because they determine whether we can go over the barriers or not. But with thermodynamics, the key is, well, thermodynamics and stability, that's thermodynamic stability in particular, that's what we get to in the limit of infinitely long time. And no matter how a barrier is, as long as it's finite, you will eventually get over it, but it can take astronomical times. So thermodynamics, low regions in the free energy landscapes, kinetics, high free energies. Oh, sorry, that was number 2, my bad, it helps to read. So what is most important for kinetics in terms of understanding things? It's related to the second one, but it's not entirely. How do you determine kinetics or a transition rate?
The rate is depending on the barrier.
It depends on the barrier. How does it depend on the barrier?
It needs to overcome it.
Right, and that means that what type of expression do you get? Right, so you need, the reason why you get an exponential expression is that to get over a barrier, you need to wait until by chance your particle, well, by distribution, your particle has that energy, right? And that means that the probability of going over these barriers is governed by a Boltzmann distribution. And that in turn, the derivation I made, what was it, on Monday this week, that you get this prefactor that there is some characteristic time that we can't predict exactly what it is, and then multiplied by an exponential of the free energy barrier. But it's because of the Boltzmann distribution we get that. And that's important because not every single exponential you will be seeing, not even in this course, it might look very similar to Boltzmann distribution, but just because something walks and quacks like a Boltzmann distribution doesn't mean that it is. But this one is. So should we you know what, since I didn't put any numbers, let's just continue down. What is the point of separating initiation and elongation free energies, and roughly how large are they for a typical helix?
The point is that the initiation free energy represents the barriers of the barrier that it has to be overcome to actually initiate and form a helix, while the elongation free energies are not that relevant.
Exactly. So this is not really initiation and elongation. This is not some fundamental physical concept. an alpha helix of any given length has free energy relative to being in coil, right? And it's up to us or you how you define that free energy. But by trying to separate that and what is the high term that you will have to pay before you form it, we know that that corresponds to a barrier, and then there is some additional extra term that makes you benefit more and more the longer it is. It's a completely arbitrary separation. It's just that this separation enables us to identify this initiation. So roughly how large are those terms for a helix? And what sign?
The initiation is positive, so you have to pay,
Yes.
and the elongation is favorable for the formation.
And which one is larger magnitude?
So as a single one, the initiation is larger, but then after
Right.
many steps of
So that
formation,
it takes a while to gain. And that leads to friend of order, then also something else. But in that case, the more residues you add, the better it is. So why don't you have it? Why aren't most helices hundreds of thousands of residues long?
in real proteins, there is always some kind of residue that will stop the helix at some point.
Yes, and that comes down to probabilities, right? You only have 20 amino acids, and you can, this is pretty much statistics, pick out the number of amino acids that are very strong helix formers. They might correspond to 1/3 of the amino acid residues, or at best 50%. 50% it might be easier. So that probability can happen. It's 0.5 raised to 100,000. That's a relatively small number, so we can kind of forget it. So this is quite true, but you also have to factor in the probability whether you have residues that even favor helices in the first time. And of course, if you put a prolin here, what would be the elongation free energy be for a prolin? That would be positive, right? The prolin doesn't like to be in a helix. So at some point, this is just for residues that actually like to form a helix in the first place. And that's why it's not always going to be negative. So how are numbers like that determined? Right, so in this case, CD spectroscopy. And this is really, I think that we have had 1 or 2 slides when you try to argue that based on these simple energies, what is the number of residues that you expect to have helical in a typical coil segment, right? The reason for doing that is that the fraction of residues that are helical I can measure in CD spectroscopy. I can't see which ones are helical, but I can see that 37% is helical. And then I can go backwards, because that was expressed in the free energies. And if I know the number of residues, I can go backwards and get the initiation and elongation free energies. So that's frequently why you do these very simple models. It's relatively hard to say that based on the shape of a curve in a CD spectrum, what is the energies of the underlying process? At least I'm usually not smart enough to do that. But if you create and model a very simple process, and you can say, what would my model lead to? The lead to part you can frequently observe, and then it's a matter of solving the equations to get the original energy or probability or whatever you were looking for back.
One question.
Hmm?
So in CD spectroscopy, how do you actually determine the percentage of residues?
Oh, so that you have, the point, you have 3 curves, right? So you have one type of curve that is the shape you would have for coil. You have one type that we would have for an entire alpha helical sample. And then you have one type you would have for beta sheet. And then you pretty much fit it. So you assume that the curve you see is a curve with 3 components: alpha times alpha helix, beta times beta sheets, and then you could say C times coil, but it's not a completely free curve, right? So that you only, the mix of 3 components leads to that you have 2 factors here. So from that, and then, so we just assume that they add up linearly, which is a fairly good approximation.
No, so I mean, I know the machine that measures like polarized light and stuff, but I was wondering, so how do you know if it's 37%?
You don't get a number from the machine, you get a curve from the machine, right? And we know what the curve should look like for a pure coil, and we know what it should look like for a pure helix. But it's not obvious that the curve should always be a linear interpolation between those. But for CD spectra, that works very well. You could imagine that the coil and the helix interacting in some nonlinear fashion, but it doesn't. So what type of transition is alpha helix holding?
Not a phase transition.
Exactly. It's a cooperative transition, but not phase transition. And that brings me back to 2 other things that I haven't told. So what's KT? Or in kilojoules? 5. Yeah. Know those by heart, and it's not because of this course. That will help you a lot throughout your career. Just having gut feeling. When do things happen? When don't they happen? What's the energy for hydrogen bond?
0.5.
Sorry?
5 kg.
Yeah. Sorry, I thought you said 0.5 for a second. That's fine. And that varies to at least within a factor of 2, but it's not 100 and it's not 1. Also one of those things that will help you tell quickly whether things are realistic or whether they happen or not. Roughly how long does it take to fold a typical helix? Yeah, or 0.1. I would say microsecond is fairly slow. It's fast. The point is it's so fast that you're entering the regime where most experimental methods have a really hard time tracking it. And you even saw that in some of these simulations I had, that it happens so fast that first you don't see anything and suddenly you see the entire helix. And how is beta sheet folding different from that, in particular from a kinetics point of view?
It is a phase transition
Why is it a phase transition?
because it's all or nothing. You either have the beta sheet or you don't.
And it has barrier on
Yes, well, you have a barrier for the alpha helix too. So the barrier is not specific to the phase transition. The problem with this phase transition is that the width of the region where in energy or something where the transition happened will eventually go to 0, right? You will have a very rapid transition. But it might take a very long time before we get there. You could also argue it's not critical to know that beta sheet folding is a phase transition, but there are many of the way we see beta sheets, whenever we see beta sheets, you usually see much larger regions of beta sheets. And that has to do with these things we mentioned. Beta sheets are not really happy to coexist with another structure locally. An alpha helix is perfectly happy to have helix and then a small coil and then a small helix and then a small coil. But for beta sheets, that is fundamentally unfavorable. And that's, as you're going to see today, beta sheets usually prefer to be in other regions of the protein than the alpha helices. And we also showed yesterday that it turns out to be an exponential dependence on the stability of the residues here. So beta sheet folding is also different in the sense that it spans several orders of magnitude. It can be anything from, say, a millisecond up to weeks or so. And that's also why you end up having these prion proteins misfolding and everything. That's virtually always beta sheets. And then I also, the next one too, sorry, how long does beta sheet folding typically take? We talked a little bit about the coil and how the end-to-end distance varies with the number of residues, either in a coil or in a DNA or something. And to first approximation, how does the end-to-end distance vary? You don't need the formula. You need, what is the power?
2.
A half. It's a square root, right? So if you're doubling the length, if you're moving from 100 residues to 200 residues, the average end-to-end distance is going to be 1.41 times larger. And that is the same thing as the diffusion properties you saw in the lab, that the square r goes as the square root of time. Or conversely, r squared goes as time, right? This is important not just for protein. It's going to be very rare that you study the coil, but there are a number of experimental methods that rely on sorting or finding things on gel based on their size. And that's why in all those cases you're going to see a square root behavior normalization in the curve. And that's because of this. And then you can show that when you take account that the chain can't cross itself and everything, you get a slightly larger number, but that's a surprisingly small correction. So when it comes to real biomolecules, in contrast to the very simple ones that we've studied, what typical terms do we need? That was the stuff that I spoke about after break yesterday.
This is the volume, so like the Leonard-Jones.
Well, so the first point, those are the individual terms, but what's the whole thing you call it? You need a... Yes, or a potential, right? You need a way to describe, as a function of the coordinates in a molecule, what is the energy, or enthalpy. And that in turn contains several different terms. Lennard-Jones is one of them. What's a Lennard-Jones interaction?
That's the attraction between the atoms.
There's both the attraction at long distance that you have for any particles and also the repulsion at very short distance that they can't cross. Good, so that's one of them. There are at least another 4.
Angles between bonds.
Angles between bonds, yes.
Proper angles between planes.
Yes, sorry, impropers you usually call them, out of plane torsions. That describes, for instance, there are many groups. So let's see here. A carboxyl, for instance. So you might have a C, and then O, and then OH. That carbon should actually be completely planar for chemistry reasons, the way the electrons are organized. So then you apply a potential to make sure that this one stays planar. We also had the bonds, right, since you said angles. And there's one thing you haven't brought up. Electrostatics. Electrostatics is actually complicated because electrostatics is painful in that all these other interactions decay very quickly, but electrostatics goes as 1 over R. And it turns out that you can't really cut that off or say that if you're far away it doesn't matter or something. It's a fairly deep, no, I wouldn't say that deep mathematical result. But if you have the function 1 over R and integrate that, you get the logarithm of R. And the problem, so that if the interaction is 1 over R, the sum of all these would be somehow proportional to the logarithm of R. And if you then make a cutoff at any distance, say 1 billion miles, the integral of ln R from 1 billion miles to infinity is still infinity. So no matter how far out you make a cutoff, you still make an infinitely large error. The reason why this works in practice is that we don't have free charges. You might think of an ion as a free charge, but you can't give me a kilo of sodium ions. Sodium ions only occur together with, for instance, chloride ions. So that we don't have free charges in nature on macroscopic scales, and that effectively gives us dipole-dipole interactions. So there are a couple of tricks that you apply in simulations that you borrow from crystal, solid state physics and crystals really, so that you can sum up an infinite number of periodic copies of these interactions or something. The only reason I'm mentioning this is that Björn and Dari, when you start doing a small simulation, they might mention something about using a special algorithm called PME for electrostatics, and that's the special algorithm. And then I spoke a little bit about energy minimization. So what is that energy minimization does and what it doesn't do, in our case at least.
You can kind of refine the model, but it won't do that much.
I would even be careful not to use the word refine. So by refinement, we usually mean that we get something that's substantially better, in the sense closer to the native state or something. And that's not going to happen because the energy landscape is way too rugged. So what the energy minimization does is that at best it gets us to the closest local minimum. It will not on average move to significantly better parts of the energy landscape. The reason why we do that is that if we're unlucky when we start a simulation or something, we might be at the red, at the horribly bad part we're starting from, and then things will crash just because the everything is going to be too bad. Forces are too large, acceleration is too large, velocity is too large. So the point of energy minimization is really to get away from colliding atoms and things like that. But the second we can run a real simulation, that's much more efficient. Which connects us to the next question. So what does a simulation do?
Explores the energy landscape.
Yes. And in particular, by exploring the energy landscape, you meaning what?
Exploring all the different microstates.
Or, yeah, no, I think that's a good formulation. And by explore, I like the word sample. So the point is not, while it might look at something as moving, our point is not to get a time sequence of points. You might get that anyway, you can show that things are, but the point is you're sampling things according to the Boltzmann distribution. But you already showed that you could do that with Monte Carlo simulations too. So why don't you do Monte Carlo simulations?
Because we don't have those limitations that you talked about, like angles and...
Well, it's not just that, right? That for a large protein, what's the problem with the Monte Carlo simulation? So Monte Carlo simulations will explore the Boltzmann distribution just as a simulation does. You've used Monte Carlo simulations, all your simplified models, and the reason we use the simplified models is that they contain conceptually the same ideas as the complicated ones. So the reason why Monte Carlo simulations are efficient is that you make much larger moves. Do you remember this old movie I showed of the water yesterday? So when you're simulating a molecular simulation in the real sense and integrating Newton's equations of motion, it's wonderful in a way because, you know, since you're moving according to the forces and potential, you fulfill the Boltzmann distribution. You fulfill this criteria called detail balance. It's amazing. The curse of it is that you do this by moving in insanely small steps so that you're hardly moving at all. The beauty with Monte Carlo is that it can take much larger steps. You're much more efficient at sampling your energy landscape. But when you take larger steps, you will on average bump into things. This works in a simple system. It works in a system if you have a protein without water. But the second you start having water or a neighbor protein, you will virtually always bump into something. So that you're going to keep trying to do Monte Carlo moves, but we will never accept any moves because the motions are too large. And that's why we normally give up on Monte Carlo simulations, but occasionally it can be very useful. As we're going to move to docking at the end of the course and show how you place a small molecule in a protein, we're going to use Monte Carlo simulations. Much better than MD. So the point is that what do we then get from a simulation? That's related to the second last question. You can actually formulate the second last question in another way. This sampling is what? It's an approximation of something.
All the microstates.
But we had a name for that, the collection of all microstates and their probabilities. The partition function, right? So the partition function is this holy grail in physics that if you know the partition function, you know every single microstate and you know how likely it is to be there. The second you know the partition function, you know everything about your system. Stop simulating and just calculate all the properties. Because all properties are just going to be averages over all the microstates, and we know all the microstates. That's never going to happen in reality. So the simulation tries to approximate the partition function by at least focusing on the important parts. So that brings us to the last question. So what do we then get from a simulation? Yes, and that in turn enables you to calculate averages of observables, right? Say the free energy or the secondary structure or potentially a transition rate between a folded and an unfolded state, how likely it is for a big protein receptor to bind a drug, some spectroscopy property, anything you name it. And I'm not sure if it's obvious, but you see the analogy here that I mentioned that we had a very simple model here where we knew the energies for initiation and elongation. By using that simple model and predicting what observable we would see, we could use CD spectroscopy to get the fundamental factors back. This is exactly the same thing we're doing, but it's about 10 orders of magnitude more complicated. So we have a model in the sense that we have a potential. You have a way you describe your protein. Based on this model, we predict what you would observe, say a free energy or something. You compare that to an experiment, sorry, not a free energy in this case, how likely it is for a molecule to bind. You compare that to an experiment, and based on the experimental result, we can then go back and say, let's see, did we have a correct model for how this small drug molecule binds to my receptor? And there are two answers to that. The answer can be yes or no. So that it's a more complicated model, but this is really the modeling part of simulations. It could also be that you have a very, very fuzzy cryo-EM data, and I haven't forgotten that we should try to bring it down to the facility. And this model is simply too fuzzy to directly place your atoms. But if you can use a simulation and predict what your structure would like, you can then use that to, well, first you can let your simulation be guided, steered by the cryo-EM data, for instance. But in particular, you can say, based on the data you see in the microscope, does this seem to be a reasonable model of my protein? That was all the questions we had. I think it's fairly good to go through them because I realize, I'll tell you a little secret. I've talked about these things before. So I know it fairly well, and that's why it's so easy for me to rush through things. And then I realize when we talk here, there are some things that I feel that I spoke about 2 times yesterday, and I realize you haven't caught them quite. So the one thing, if you have a chance, try to answer questions, all of you here. If you don't know, that's an even stronger reason why you should speak up. The worst thing that can, again, I will spend as much time on those as we need. Absolutely worst case, I'll start recording all lectures offline so that you can look at them yourself. Today, we're going to look at real proteins. Lots of pictures, very little mathematics. And as you might have guessed by now or even know, that the recurring theme here is really how we create these complex assemblies from simple building blocks. I'm going to talk a little bit about something called supersecondary structure and motifs that you might or might not have heard. And then we're going to talk a lot about some classes of proteins. In general, in the world, I would argue I and the book and a whole lot of other people would argue that there are 3 big classes of proteins. And now I see the slide and I realize that I've drawn their size in exactly the opposite order from what they would be. The first type of proteins we determined or found and realized there were proteins in that sense and got structures for were globular proteins. Globular proteins are the vanilla, water-soluble proteins. When you think protein, this is usually what you think about. The reason why they're called globular is that they're typically small, they're very close to spherical, simply to minimize the surface of interaction with residues with water, right? Because you have hydrophobic stuff on the inside. They perform a ton of functions in your body and they're very easy to work with. I wouldn't say easy, but they're possible to crystallize. That is also why most of the known protein structures are globular proteins. Not necessarily because most proteins are globular proteins, they probably are, but it's were the ones that we found easiest to work with in the beginning. This is a membrane protein, which is actually larger than the first one there. Or no, it's probably about the same size. Membrane proteins are still relatively rare in databases. It's a matter of a couple of hundred membrane protein structures. And the main reason for that is that they live in this lipid fat surrounding, and it's super hard to crystallize anything in fat. Actually, I would argue that it's typically impossible. So there are 2 ways people get membrane protein structures, either by putting the entire protein in sort of micelle, or taking the protein and binding it to an antibody, gigantic antibody. And then you can crystallize the antibody, and then you essentially get the membrane protein in your micelle as a small cargo to the antibody. And then you just happen to see that in the X-ray structure too. There have been lots of Nobel Prizes for membrane protein structures the last few years, and I bet we're going to see more of them. Roughly 1/3 of proteins in your body are either in the membrane or they're connected to the membrane surface or something. So they're much more common than you might think from the number of structures in the data banks. And I have to confess that I'm not entirely unbiased here because this is my life, essentially, membrane proteins. But one of the reasons why they're cool is that they're very functional. They're doors and windows, they pass signals, they very concretely do something very biological in the cell. And then we have on the right here, we have the type of proteins that's pretty much the opposite of membrane proteins. These are boring proteins, I would argue. Large fibrous proteins, so that these are extremely large structures that form bone, teeth, hair, skin, and everything. The special thing that they're so large that you can see them. And there's very boring, simple structures that just repeat or something. There are a couple of cases that can be fun to see. So I'm going to try to take you through a bunch of these. Membrane proteins, I'm going to spend all of tomorrow talking about. The book is completely outdated on membrane proteins. I'm going to deviate completely from the book there. And today I'm going to start talking a little bit about fibrous proteins and then about globular proteins. I very much doubt that you're going to do much work on fibrous proteins, but there are a couple of things that I want you to have seen at least. So take the fibrous protein part with a grain of salt and think most about the globular proteins when I go through them. So all these structures, both the structures that I draw and other structures, are actually not proteins, right? But they're models of proteins. And there are a couple of programs that, in principle, we could make a lab on this if you want to, but I don't think it's worth it. There are some free programs that you can download so that you can actually look at proteins. The proteins you get from the protein data bank is just a horrible, simple text file where each line says atom, and then it says what the number of the residue on the atom is, the name of the residue, the name of the atom, and then XYZ coordinates in angstrom. you can't do anything with those files. So to be able to ever look or investigate proteins, you need to download one of these programs. VMD is one of them. PyMol is another one. You can just search for PDB viewer or something. The reason why I still want to stress the importance of these programs is that a protein like this, the ribosome or something, that can have like hundreds of thousands of lines of atoms. And just being able to understand, forget about the computations and simulations, just being able to understand what these do and what parts you have it's a bit of an art to learn how to represent that well. So this is hemoglobin, which is the oxygen carrier in your blood. The small green group you see there is what you call a prosthetic group. And a prosthetic group is a group that is not really, this is not an amino acid, but it's a group that exists in your blood or cell, and once the protein has folded, this group binds to the protein. And do you know what this particular group is called? Yes, kind of. It's actually called, there are two names. It's a protoporphyrin. And when a protoporphyrin binds an iron atom, it becomes a heme. And right next to that iron atom, you can bind the oxygen. So you have, and hemoglobin actually consists of 4 subunits. So you see 2 on this side and 2 on the other. You would not have seen that had I shown you that image. And that is the image that you see by default, right? So that to be able to visualize and understand proteins, you need to learn a bit to work with chains or surfaces or coloring, different ways of drawing them. That is really what the bonds and the atoms are. It's just bonds and coloring by atoms. You can decide to draw the main chain if you want. So that's just essentially just the alpha carbons. Even that I find very hard to understand. Or you can actually draw all the chains. So here we've drawn here we see that it's alpha helices, and you might be able to start to see that there are 4 different subunits, right? You certainly don't see that up there. And you can draw all the side chains, but here you see the curves too. The more information you have, the harder it is to see the trees for the forest. And eventually you might want to zoom in on the protoporphyrin and actually see the oxygen right next to it there. So it all depends on what you want to see. There are some simply stunning visualizations that are made usually with these tools, in particular where people are awarded Nobel Prizes or so for their work. And there's actually even quite a lot of research in modern visualizations on how do you light systems like this. This is a bacteriophage, and I'm not going to go through the story, the history of early molecular biology and everything, but these structures were super important when people first started studying molecular biology, Max Perutz and others. Ribosome, I think, gigantic structure too. So here the red chains here are actually RNA, while all the blue stuff is helices proteins. That's also not something you would see unless you try to visualize it correctly. Or you can use it to simply do a visualization or simulation. What happens if you have a DNA and you're going to pull it through a nanopore? So this is not something, this is a device that doesn't really exist yet, in particular not through a graphene nanopore. But people are, of course, running simulations here. So why are you running simulations before we even have nanopores? Right, because the way we're going to try to detect this is that based on what base pairs you have here, we're going to see hopefully the bases here have slightly different composition, right? So hopefully there will be some sort of difference we can detect, say the capacity or the some current in the graphene layer or something. And if we can see if that is not possible to detect even theoretically, it's kind of a bad idea to start building the pores. The cool thing is that you can show in simulation that it is possible to see the separate signatures for A, G, C, and T here. That doesn't mean that we can discover it in an experiment, but that means that it's at least worth trying. And that's what a lot of the groups in the world are working on right now. So this might fail, we don't know. But right now there is at least one small company called Oxford Nanopore that has manufactured a device that is not based on graphene or anything, but we're starting to have the first so-called nanopore devices for sequencing come out on the market. Have you seen them? They're super cool. Unfortunately, I don't have one here. They're the size of a USB stick. Actually, they're not just the size of a USB stick. It is a USB stick. So that this one is the type connected USB. You open it up, you put a drop on it, you close the lid, and then you run it. So it's a sequencer connected to your laptop.
I think we saw when we did the satellite run.
So that and people are still a bit worried because it's not entirely obvious how you create the sequence, right? That do you start having some systematic errors because it's a completely new type of sequencing technology. But in many of these cases, just being able to visualize it and showing what happens, this is more connected to simulation than you might think. Because on the other hand, it is a bit dangerous because it's so easy to be seduced by this beautiful plots, right? But this is just a model. It's not reality until you simulate it and show that it matches reality. So the first of these classes that I'm going to speak to is fibrous proteins. The reason why fibrous proteins exist is that they are the structural building blocks of your body. Otherwise, you would just be a small amoeba or something with, well, not amoeba, a collection of amoeba, multicellular, but you would just be a pool of cells. And most of us aren't pools of cells, and that's because of these rigid building blocks. That in particular means that it's pretty common that they don't really have a direct biological function. Their biological function is really to act as the scaffold. They are usually hard, very hard, and to be able to create something that's millimeters or centimeters thick, you can't have one protein structure that way, right? So that they're typically built in a huge number of hierarchies. So you have 1 helix, 4 helices, 8 helices, 16 helices that go together and together and together and together, and eventually you to get to levels where you can form bone, tooth, nails, etc. Shells, claws, that's all proteins. So that the protein itself is large, but assemblies of these proteins are even larger. So it's very common to have things like this that are long threads or filaments or chains or something, and then you go to larger and larger and larger scales. I think that is from bone, actually. And they're assembled in very simple ways, normally just hydrogen bonds. Let's start with the very simple ones. Silk fibroin, one of the first ones. This is actually what gives silk its properties. And it's just beta sheets. You felt beta sheets, pure beta sheets. And it's serine, glycine, alanine, glycine, alanine, glycine. And then you repeat this, serine, glycine, alanine, glycine, alanine, glycine, serine, glycine, alanine, glycine, alanine, glycine. Pretty boring. But that's the whole point. These should not have a very specific biological function, right? If you just want large extension, just keep repeating the same pattern over and over and over again. This in particular, the serine in particular, means that they have separate hydrophobic and hydrophilic surfaces, which means that they also like to form layers, and these layers assemble in larger layers, and that's why it eventually gets large enough that you can see it. Then, of course, in real silk, this would not be entirely pure, but it's kind of like a crystal, but still not quite. I'm not sure whether I have this here. It's either here or in another slide. Have you ever looked at a shampoo bottle and see that they say that they contain silk protein? That has absolutely nothing to do with silk. It's this. And you think that it's something, well, they announce it as if it would be expensive or nice in any way. That first, it has absolutely nothing to do with your hair. Your hair, for instance, whatever, it's also going to be protein. Your hair will not spontaneously start to absorb other amino acids. There are other properties in the shampoo that makes you get, well, shiny hair, whatever it is. So it's a complete market scam. It has absolutely nothing to do with silk. And you can manufacture tons of this at no cost whatsoever. It's the cheapest amino acid you can imagine.
Can we make silk out of it? Like
No, because then you need these very long, very specific structure that would be completely different. So getting the large structure right would be hard. It's actually good. I bet you can make artificial silk, but it would probably be more expensive. Silk is not that expensive. Silk is expensive in the Western world once this has gone through 14 different merchants and everything. But the actual farmer with the silkworms and even silkworm silk isn't that expensive originally. So I would assume that's why it's not a gigantic industry. Way more money to be made from cosmetics or something that just pretend that it's expensive and sell the cheap stuff. Collagen. Collagen is really screwed up. So collagen is a helix. It's actually 3 helices. Do you see that it's proline, 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 proline? You would not believe that you would see proline in a helix, right? Note that it's not an alpha helix. There is not a single hydrogen bond inside each chain here that is either white, gray, or black, but there are hydrogen bonds between the chains and frequently with the water. Very strange structure. This is a quarter of the protein in your body. Very boring protein. Bone, teeth, skin. Just creates these large extended structures, and it can be either very hard or a bit brittle. And this can be like 300 nanometers long, extremely long fibers. And then it aggregates into larger and larger and larger layers. This consists of mostly proline and then a little bit of glycine. So it's the glycine here between it that gives the link so that it can actually turn, but it's 2/3 proline. Almost all the protein in your body is in collagen. If you wanted to go into detail, these chains are actually not strictly identical. There are 2 types of one chain and 1 type of a slightly different chain. It's not super important. But have you seen what we've done here? So we've taken our chains, and I think we've assembled 6 of them or something here. And this is very typical for fibrous proteins, that you go from one large chain, and then you make a chain that's even larger, and then you have several of these filaments that are even larger. And eventually you're going to get to something like this. So this is dentine in your teeth. And I should see that this is actually an electron microscope image, and I think it says 100 kilo electron volts there, and I should know what the extent of that is, but I actually don't remember. But this is pure protein. But the other thing that can happen, remember that glycine? If that glycine is mutated to any other residue, it's no longer going to be as flexible. And when that is not as flexible, you're not going to get all the beautiful hydrogen bonds in these structures, and then you're going to get brittle bone disease. So it's a very classical example of a single residue mutation that leads to pretty severe disease, and it's all based because it's influencing the structure. Remember this thing I said early on in the course, that what is the central dogma of molecular biology?
Sequence, structure, function.
Yes. So that's like sequence mutation leads to a problem in the structure, leads to a problem in the function.
So is there just one sequence for an individual collagen, or are there multiple types of collagen?
Normal collagen is pretty much just one sequence. And again, you might have some very rare differences that every 100 residue you might have something else. But the reason why I can show you these sequences is that there is extremely little sequence variation, because there is no point, right? If you look at hemoglobin or something, and I will get back to that, there might be reasons to have slightly different properties of hemoglobin, but bone is bone is bone. We don't really have needs for different types of bone. There is no point in natural selection.
Collagen is not a ribosomal protein, or
No, I would be almost certain that it's not assembled in the ribosome. That's actually a good question. I don't know that.
because how these mutations actually happen.
So this mutation, this is usually something that you're born with. This is something you're born with. Most mutations, in rare cases, of course, mutations happen spontaneously, but then they happen in one cell. That's usually not a problem for you, unless this is a mutation that causes this cell division process to go berserk, i.e. cancer. And the problem with cancer is that that one cell is then multiplied into billions of cells before you know it, and eventually it will take over your entire body. But in general, the body deals with mutations by killing that cell. You have plenty of cells to spare. Alpha helices in general, remember that I said that collagen was really a set of helices that assemble into more helices, and this happens quite a lot. Technically, I'm not sure whether I would say that this is a fibrous protein, but this whole point of having many alpha helices go together and building even larger structures, that is something that recurs in lots of places. So in every single, remember that I spoke about titin in one of the first places. So there is a protein called myosin in your muscles, and that's this protein that is this coiled helix here. And what happens when a muscle contracts or something is that you go through a process where this protein essentially walks on this other muscle fiber here, and that's how they contract in a millisecond or something. They're using a huge amount of ATP. There is a ton of different structural transitions they go through, so there's no intelligence here. It's that they're just going to something that's more favorable thermodynamically. But it's easy to understand these things when you see one protein. I'm still simply amazed the way the body can handle these things purely by thermodynamics. It is only thermodynamics. Very good question. The reason why they're typically 3.5 residues is that after 2 turns you want to get back, because otherwise you would keep shifting it a bit. But that's not always the case. So that's, in many cases, even when you have these coiled helices, the entire coil tends to move around a bit too. It's not important, so I'll skip that. And that brings us a little bit more to helices. I'm sorry, helix geometry. Why am I going to say this? The reason I'm going to say this is because this will be super important tomorrow when I talk about membrane proteins. If you just look at a helix that way, it's not really going to be too obvious. You can also look at it as one of those cartoons, but that doesn't really tell you a lot either. Or we can look at the surface of the helix. And if you look at the surface of the entire helix, in particular if you see this in a program, you will actually see that there's kind of a valley here, and then there's a peak here, and a peak here, valley here. So there's kind of, depending on how you look at it, there are both valleys here, and there are peaks between the valleys. And these peaks, of course, correspond to the side chains. And depending what peaks you look at, well, the side chains are points, right? There are kind of two peaks, one around 40 to 50 and one around minus 20 to 30 degrees. What do you think can happen when two helices interact?
They kind of interlock.
Exactly. Or that depends on your residues, of course. What if you now put two gigantic residues, say two tryptophans, right next to each other? They're not going to be able to pack, right? Because, well, even if it's not repelling electrostatically, it's simply there isn't enough room. So that ideally, if you take one helix, move on top of the other, and then rotate it a bit, these can pack beautifully. If you just look at all your protein structures, how common is it that you see two helices packed that way? No, you'd never see it. They always pack that way. This is the reason, because it might sound stupid, why on earth aren't they really packed? And the reason why they almost make an angle to each other, and there are two angles they can make, either roughly 20 or 50 in the other direction. And the reason for that is exactly this. Otherwise, you would not be able to pack their surfaces very well. Francis Crick predicted that 53, 10 years before the structures. Pretty cool. Yeah. They kind of had he had a productive year. You see that, and there are several helices. In particular in coiled coils, this is important. So when these helices cross each other, first it's really good to have glycines, because glycines are small and they don't really take a whole lot of space. But it's also very favorable to have these red residues be leucines. So why would that be useful?
They're also quite small.
Sorry?
They're also quite small.
They're hydrophobic, right? They're both small, but they're also strongly hydrophobic. So it would be bad to expose these side chains to the water. They love to interact with each other, on the other hand. And when these are placed roughly every 7 residues apart, they form a beautiful, we literally call them leucine zippers. Because it's a zipper that you can zip up a very, very long. If you just saw an alpha helix, and these helices can be like 50 residues long, you're not going to see them in the protein data bank. Why aren't you going to see that in the protein data bank? Well, no. So these frequently occur as part of a receptor or on a bacterium, something sticking out of a cell. So they're long, they're fairly floppy, and they're extremely hard to crystallize, right? If they're 50 or 100 residues long. So you're never ever going to see them in a crystal. But this is the great advantage with bioinformatics. We see this with sequence prediction nowadays, that there is something beautiful that's alpha helical. In addition to alpha helix, you see that every 7th residue is a leucine. And by that time, you know that you're going to have a coiled coil leucine zipper. Roughly 10% of these residues are cysteines. So how can you use that? Well, I kind of said it there already, right? So if you now add disulfide bridges to this, you can make this even stronger. Remember that for a second. Because this is alpha keratin. It's hair. All your virgin acts is not entirely true. There are some other trace amounts of other proteins in hair, but hair is almost entirely alpha helix. So what you do is that you have an individual helix that first coil up in a coiled coil. So there are 2 of them, and then you have a pair of these 2 ones, And then depending on exactly how these intermediate filaments or something pair up, you first form a matrix and you form a macrofibril, and then you eventually form these very large structures that actually is the hair, which in turn connects to the cortical cells and the cuticles in your skin. So I actually forgot exactly. You can actually, you know what? That's a very good exercise. You can calculate how many helices there is in an average hair if a hair is in the ballpark of 0.1 millimeter. But then I realized that I actually looked up how fast hair forms and how many alpha helices that correspond to. And I kind of lied to you yesterday. I thought that this would be almost at the limit speed of what alpha helices can fold, and it's not. Hair folds roughly 1,000 times slower. Why is that? It actually makes sense if you think about it. Well, we need to build all the amino acids, right? We're limited by the speed by which it can provide new amino acids to the cells and everything. So in this case, we're not limited by the alpha helix formation speed, but it's still pretty decent, right? You're adding something like every 30 milliseconds or something. You're adding an amino acid to each helix. So this brings us to some other question, the cysteines. What could you do with the cysteines, you think? Oops.
Place your hair in some sort of strange, curly fashion.
Or a permanent wave, right?
Yeah.
That's how you make a permanent wave. So that normally in the hair, those cysteines will form disulfide bridges. So now you first add a reducing agent, so you break all your disulfide bridges. And then you form the hair in whatever shape you want it, or just comb it straight. And then you add a new oxidizing agent, so you reform the disulfide bridges. And then the hair will tend to stay in that. The hair is still certainly flexible, right, and everything. But so for instance, if you want curls in it, then you now force it to form the disulfide bridges in a form where the hair is curled. Very simple chemistry. Sorry? I normally don't, but if you go to the hairdresser and get a permanent wave, they are working with your disulfide bridges in your hair. Yes, that's how it works. I think it was more popular in the 80s than today. But Elastin is another strange protein. It's a highly elastic fibrous protein, which is kind of similar to collagen, but it's a bit softer. And what happens is that you're having these fairly short collagen-like helices, and then they're cross-linked by small leucines here. And this makes it very elastic. You can stretch it and pull it. This is what nature uses in things like your aorta. And many of these diseases, what you get when you're older is that, for instance, if you have a deficiency in some of these lysine-modifying enzymes, eventually these protein will change so that it's more brittle and it's not as flexible. And that's, for instance, why you can eventually get an aorta rupture or something. It's very rare to have an aorta rupture when you're 20. But eventually, as you get older and older, your vessels becomes more brittle and fragile. This is incidentally related to something. Did you read the scandals about Paolo Macchiarini at the Karolinska Hospital? That they hired this Italian surgeon who kept inserting artificial trachea and everything? Long term, this is of course the direction in which the entire field would go, that using biomaterials instead. So what is the advantage of having a biomaterial instead of a plastic aorta?
Don't have to worry about lysine modifying.
Well, lysine modifying enzymes aren't going to work on the plastic either. So that's not a problem. What's the problem with plastic?
Body will reject it.
Right. Your immune defense does its job. What happens if you have a protein? Yeah, it's biocompatible. So there are, of course, some metals such as titan or something that are largely biocompatible, but proteins is even better. Proteins are awesome. The body won't even try to reject it. It's still not something that's used a lot because it's fairly early research and it's hard to make these things. Again, nature has had 4.3 billion years to perfect these processes. But long term, if we could make things with biocompatible materials, you could replace organs or anything, but people still would not be dependent on anti-rejection drugs, which is frequently the reason why people die after these procedures. Because if you take these anti-rejection drugs for the rest of your life, suddenly you die from a cold, not your new heart.
Could you design some plastic that would have a thing that is imitating antigens on the surface, so then it would not reject, the body would not reject.
So, well, the problem is kind of the antigens that the body is reacting on, right? Or when it's biological, I don't think it would be so much antigens. It's hard because we don't know exactly. So that had it been a biological material, then it would usually have been proteins that your immune defense reacts to, specific proteins in a foreign organism. The reason why this would work is that it would use the same type of protein as we have in a human. There are some materials that appear to be more biocompatible than others. For instance, titan. So that's what you frequently use in hip replacements or something. For whatever reason, it's a very smooth surface. It doesn't appear to upset your immune system a whole lot. So there you won't really need an anti-rejection drug. Plastic is just as complicated as proteins in a way, right? There are billions of ways to create these polymers. Plastic is nice in many other ways. You can shape it. Long term, you can 3D print it. And people are actually using that, not so much plastics, but when you perform surgery in your brain or something and you remove a piece of the bone, then they actually 3D print small metal cages to be part of your skull. So in principle, the answer is yes. It's just that it's a very young science. The other problem with all these things is that it's very rare that you perform surgeries on healthy 20-year-olds, right? So then, and if most of your subjects are 85 years old, there will be lots of deaths, not necessarily because of the procedure, but because these patients were very ill in the first place. So I think that's all I'm going to say about fibrous proteins. And now we're going to get into much funnier stuff. Globular proteins. This is going to be biological. So biological. There are, you know about the alpha helix and the beta sheets. You could probably almost guess this. There are proteins that are only alpha helix. There are proteins that are only beta sheets. And there are some proteins that are mixtures of helices and sheets. And that's pretty much the way you divide it. Have you studied anything about folds and anything in the bioinformatics course? A little bit? Some of you might not have. So let me know if I'm repeating some stuff you already know. Otherwise, I'll take you through it. Here, too, in some way, you would like to try to classify things. But it's an extremely large space. And what you might not think about, but what we're doing, in the very first lectures in this course, I was studying atoms, right? I was studying how is the alpha carbon connected to the nitrogen, and how is something else connected to the hydrogen, and how is that connected to the beta carbon. After that, we started looking at how amino acids are connected to each other. So we are essentially gradually moving up in some sort of sequence space from primary to secondary to tertiary structure. And the idea of doing that is that, just as you saw yesterday, all this is really a collection of packed atoms. But understanding how the chain goes helps us to trace and understand evolution in many cases. And that is hard to understand. So this is starting to get a couple of years old, so it's not that common anymore, but it's very common to try to look at topologies of proteins. And you can, for instance, decide to draw helices, look at this from the top, and draw this complicated structure with triangles for the helices and circles for triangles for the sheets and red circles for the alpha helices. And then you can vary this structure, which might be complicated. Do you see the pattern here? So you have a helix sheet, helix sheet, helix sheet, helix sheets. And all these sheets, they line up together. So all these sheets are going to form hydrogen bonds with each other, right? But the actual sequence goes in and out, in and out, in and out. Here is an even more complicated protein. So that while this one helps you see the sheet, this one helps you see how the sheet is formed in order of the sequence. These types of plots are getting more and more rare, but this is the reason why we typically like to color things by secondary structure. It helps us understand in principle how things work. Of these structures, I would actually, maybe surprisingly, alpha helices don't form the simplest structure. Beta sheet structures are usually much simpler. And you might disagree with me seeing that protein, which might not be the best example. But if you ignore all the coils here for a bit and everything, these are actually very nice and simple. So you have a sheet there, you have a second sheet there, you have a sheet there. And it's not too many complications between rotations and everything. You almost never have more than 2 stacked sheets. That's a very strange question. Why don't you have more than 2 sheets? Well, we'll get back to that, but nature appears to prefer to keep things simple. You might not agree with that, considering some of the beta sheets protein we will see later and everything, but nature adds complications, but only the complication we actually need. The larger a protein is, the more expensive it is to build, the more expensive it is going to be to tear down when we're done with it. How long do you think a protein like that survives, by the way, in your body? There's a reason I'm going to ask it, because I have that in a second slide later today. Ballpark.
A month.
You know what? That's pretty good. That was like 2 to 4 weeks. Yes. You know, see, this is the advantage of guessing. If you have absolutely no idea, if keeping quiet, you're not going to have a chance. And now you get it right. The second thing, if you're going to be a successful scientist, you should just nod rather than say that it was a guess. No. Most of the sheets are anti-parallel, but there are certainly parallel ones too. And then you can start, the book goes into some detail about this. You can actually, if you're interested, you can actually, there are some statistical differences whether do you typically go this way or do you typically go that way. Why on earth do you care about things like that? Unless you're a physicist to think that it's fun in principle.
One side can be flat, so contractional.
Yes, or you can certainly think about it in specific structure, but this is remarkably useful if you're ever going to start predicting protein structures. Because it turns out some of these are extremely rare. And if you now have 2 of these blue right next to each other, but you can't see the red loop, are you going to guess on the probable or the improbable one? So if you don't know what it is, right, we know that one is way more likely than the other. That's likely going to be the structure. Let's guess and pick that one. It even turns out that some of these don't exist at all in nature. We have no idea why. It's just that we haven't seen them. Nature might be a bit late, but it will come by 5 billion years. Since beta sheets are flat, there are pretty much just 2 ways in which it packs. It can be either orthogonal or they can pack in a aligned parallel way. They get just so slightly different properties. I think, yes, I have an example. Start with the one on the right. Gamma crystalline, that's what you have in your eye lens. Your entire eye lens consists of beta sheets too. You know what, this is kind of funny, right? Because we had silk that was entirely beta sheets too. Silk is certainly not transparent. Gamma crystalline definitely is transparent. So even though things have the same secondary structure and everything, they can definitely have quite different properties. The other protein I might not have had, I'm going to show it. If you look a bit at how this protein, this moves too much, we'll have to go here. We start from the N-terminus, and then we go up here, and then we have a very tight loop, and then we go down, and then I have a very tight loop again, and then I have a long loop. Do you see that the long loop here goes over the short loop, and then we go down, and then we keep repeating this pattern, that a short loop, and then go down, and then the long loop over the short loop. That's a very, very common pattern in beta sheets. That it's so common that it even has a name. It's called a Greek key. And it has to do with these shapes, right? You see that a short loop and then a long loop, so you always have a short loop. Why do you think nature uses this pattern? Apart from the fact that it's beautiful.
Because you can use a long coil to do something, and the short term is easy to do.
I would argue, but here I am certainly sticking out my neck a bit. I would argue that nature uses this for exactly the same reason that it's used here. That's actually a very good reason too, but why do you think the artist picked this pattern?
It fits really.
You can draw it without lifting your pen. Do you see, right? That you just continue the pattern. It's a continuous pattern. You never ever lift your pen.
You could do that with
And of course, lifting your pen here would correspond to having a break in your sequence, right? Then you would no longer have one protein. Then you would need 2 different proteins. So this is a way to have a single chain pack into something that's fairly rigid. So why?
More structure than just heavens.
Sorry?
So it gives more structure.
So that's a good question. Oh, I love this. That's a good question. Right? What would be the use of just having a large beta sheet this way? I can keep drawing. The answer is not a whole lot, right? Apart from the fact that it might be beautiful to have in your wall or something. Nature needs to do something. So if you have two of these very large beta sheets together, what would the use be? Well, no, not really, because it's going to be open on all sides, right? So that's like if I take two pieces of paper, these two, it's not really going to be the world's best channel or pocket. It's just a bit too many holes in it. So if you move to another protein, this is the other type of protein. Do you see here how nature has frequently tried to close it up, either with a helix, or in this case with small loops or something, so that nature frequently tries to use this loop to close the pocket. And ideally, you would like to have something here, which you actually have here because you don't see the side chains, something that is pretty much closed on three sides, and then you just have a small opening from one side, because suddenly you have a real pocket where you can bind something, and it doesn't get out. So the reason why we want these loops, we don't necessarily want the loops that are as small as possible, but so we want to form something that is pocket-like, and then we can use these loops to close the pocket on the top and bottom and sides. So this is this fatty acid binding protein that I showed you very quickly at the beginning of the course. Here we have a fatty acid, an oleic acid, bound in it. This is a real X-ray structure of it. This is a really cool protein because it's completely hydrophobic on the inside. So it has this hydrophobic binding pocket, while it's hydrophilic on the inside. And this is the protein that transports all the fatty acids in your body so you can build your membranes. Extremely cool, small, simple machine. Kind of amazing how nature does it. But the funny thing with beta sheets is that you might think that there are so many possibilities, and in principle there are, but you don't see most of them. There's only compared to all the possibilities in the world, there's a relatively small amount of topologies that we actually observe. We never see mixed parallel and antiparallel beta sheets right next to each other. It's usually that you have a large region of the protein that's either parallel sheet or antiparallel sheet. Don't ask me why. Well, you probably know it's likely related to entropy, but we don't really know. Left-handed crossover form for sheets of these two red coils I showed you, it virtually never happens. And part of this, of course, comes from the properties of amino acids, what's favorable both entropically and in terms of energies. And at the end of the day, we have the proteins we have because to form a stable protein that is happy to be folded, you need its building blocks to be stable. So in most cases, that these building blocks are not quite as stable as the ones we do observe. But it's still very strange that this, no matter how small these effects are, you're talking about differences of 1 or 2 kcal or something. And it turns out one of the forms we never see for whatever reason. I'm going to come back to that a little bit because this enters in prediction. David Baker had a really beautiful result that I have it in 2 slides. So anti-parallel beta sheets are by far the most abundant. You can go up, down, up, down. And that's based on these hairpins we talked about. Why is that more common than parallel beta sheets, do you think?
Less sequence needed.
So the problem is that what you need to do to create a parallel beta sheet. Yeah, so once you have your first strand, right, you need to go elsewhere and create a helix or do something so that you can come back to the start, and then you start forming the next helix. And this, of course, makes it an even more long-range interaction. Then you're going to need to mix sheet, helix, sheet, helix, sheet, helix, or something. There are some structures that do that, but it's a more complicated structure. But just going up, down, up, down, up, down is so much easier. And that likely makes it faster to form, but that's just hand waving. I certainly have not proven that. The other thing is that you can't really have a loop or a knot or anything, but the funny thing, there is no rule. There is no rule in biology without an exception. So there is a small protein called pepsine that if you look at the structure, it actually, you see there how the green one goes on the outside of the yellow helix and then in again. So that is actually a knot in the mathematical sense, which is a small, very rigid protein. It doesn't matter what it do. And that brings me back to the other thing. You have actually seen some knots. When could you create knots in a protein?
Toxin we talked about.
Yeah, so how did you create the knot there?
What's the disulfide?
Right, and that's how nature can cheat a bit, right? It's very difficult. The reason why do you think that we don't have knots in normal proteins?
Because it's too strong.
Well, it's not too strong. It's something more complicated.
It's very hard to form.
Yes, because at some point you need to fold the protein, right? And that has to fold by following the normal rules of nature. And for that to fold and spontaneously forming a really complicated knot, that's never going to happen entropically. But the beautiful thing with the disulfide bridges, how do they get around that?
They don't have to fold complexly, they just form the knot afterwards.
Exactly. So the point of when you fold the protein, you don't have your disulfide bridges. You can form those once you're folded to stabilize the protein. And that way you don't, it's not a knot in the sense that the backbone in these toxins don't form a knot. So the backbone is kind of a normal backbone, but once you have folded the protein, the cysteines will lock in the backbone in a way that you can't really unfold the backbone anymore. It's pretty smart. So there are 2 more slides I'll take on beta sheets, and then let's have a break. And this leads to something that David Baker people realized a few years ago. It turns out that this is the normal beta meander, which comes to the way rivers flow. Anti-parallel, up, down, up, down, up, down, when the sheets would be in order 1, 2, 3, and 4. These are the Greek key patterns. And what I've drawn under here, that's kind of how common they are. And then there are a bunch of patterns that you pretty much never see. There might be some of these that you see once in a blue moon, but at least a couple of these have never ever been observed in nature.
Do they cross over each other?
Well, partly it's because they cross over each other, partly because I would imagine this one, for instance, right here you have the end of the sequence stopping right in a loop, and again, it's not impossible, but then you would need to have another loop and move that to the side or something. It's simply not that common. So what David Baker et al. Realized that when it comes to predicting beta sheet structure, you don't need to consider every single possible beta sheet. Because if you've never ever observed a sheet in, let's say, that conformation, this is arbitrary, I'm not saying that this one isn't formed. If you've never observed this in the 120,000 PDB structures we have this far, if you have a new PDB structure that you're going to predict, are you going to propose that structure for a beta sheet? No. You will likely propose one of those 3, right? So that you can use this, even though we don't know why, which is both the power and curse of bioinformatics. We don't know why, which is a bummer. But we can see the writing on the wall, or the whiteboard in this case, that if something hardly ever occurs in nature, it's with all likelihood, it's going to be extremely unlikely that this occurs in our protein too. So suddenly you can weigh the probability with which we predict different things based on how prevalent they are in nature.
So do we have to wait for an X-ray structure then to see if the predicted structure is right?
At the end of the day, that's the only thing you can do. But there is something else you can do. You can be fond of laws and obey the laws of nature, or is there something else you could think of trying to do? Well, or can you try to design a protein that would be stable in that sense? Nature hasn't done it yet. But just because nature hasn't done it, doesn't mean you can't. And people have actually been successful in that. So you can create structures that nature has not yet created. And this becomes complicated because that is a structure. Will you ever be able to predict that structure with bioinformatics? And that's not really a shortcoming of bioinformatics, right? Because bioinformatics rests on the assumption that evolution works, that we find patterns in evolution. But if this pattern is not present in evolution, you've lost. And that's why big programs in bioinformatics, for instance, like Rosetta, they tend to use a combination. Homology modeling is insanely powerful when it works. You should always use that first. But there are, of course, cases where you have to go back to physics and start to optimize these things and see, can you create a stable sheet here? So in this case, this is still the last 5-6 years. So this is on the toy level where people are trying to see whether they can build something that's stable. But today I would say that there are probably 24 examples in the literature where people have built proteins that nature, in completely new folds that nature hadn't used before.
And it will be functional then?
So that's the other question. What function? What function do you want?
For example, if you are designing a drug and you want to create
Yeah.
more stable
So I thought of bringing this. Okay, since you asked, I'll take this up. Because this is, of course, the really good. Here I was just talking about designing structure, right? But what's the point of designing structure? Well, it's, of course, a point of designing structure just to see conceptually, can we do it? But the goal is to design function. Do you think anybody has done it yet? They have. Even David and others. People have designed enzymes. So remember, an enzyme is a catalyst, right, that speeds up a reaction. And people have not just designed, taken a fold, a protein that is not an enzyme, and turned it into an enzyme. That was not so hard. But what people have even been able to do, they've been able to find a chemical reaction that is slow, and that there is no known enzyme that speeds up this reaction. And then design an artificial enzyme to speed up a reaction that was previously not catalyzed in nature. And the cool thing is, of course, if you can start doing this for important biotechnology processes and everything, there are insane amounts of money to be made from making these processes go faster. So this is certainly happening a lot in biotechnology today.
Just to make things faster.
Yes, and that's part of, based on a tradition which, whether it's healthy or not, doesn't matter so much. The problem is, there is a general problem in life science today that is getting the requirements for putting a new drug on the market or performing clinical trials, they're getting harder every day. Everybody wants a drug, but nobody wants to pay for it. It should be so safe that ideally you should test the drug for 15 years before you put it on the market, and after 20 years your patent expires. So that's why it's getting, I would argue that the way we have currently developed drugs is heading towards a failure. Or you can target biotech industry. Like 12 months after you have your new protein, this can be being used in a company. Because you're producing soap or something, it doesn't matter. It's like you're not administering this into humans, right? So in biotechnology, you don't necessarily need an ethical permit or anything. Just start doing it. Use it in the factory right away. Now, the reason why we still have pharmaceutical companies is that, in general, people are willing to pay slightly more for a cancer drug than for soap. But that's why there's so much work in biotech today. It's easier to sell it. And that brings me to the, I think this is, yes, the last slide on beta sheets. Sorry. Beta sheets have this funny ability that I mentioned this yesterday when we talked about prions and misfolding, that you can have two beta sheets dimerize with each other. And at this point, it starts becoming a definition. Is this one or two beta sheets, right? Effectively, this is one sheet. Okay, this one goes up and that one goes, this one goes down and that one goes up. So it's just a continuation of an anti-parallel beta sheet. And this leads to extremely strong dimerization surfaces. You can just start counting up the hydrogen bonds. This is important because this is also a way you can start to design protein-protein interactions. At the end of the day, a protein-protein interaction is nothing else than two proteins binding to each other. And you can sit down and try to design something, make two proteins bind each other better. Or in many cases, if you're developing drugs, it turns out the protein binding was causing something to activate a receptor. Assuming, in this case, it's a dimer, but could you imagine doing something here if this is bad? Could you imagine doing something to break up this interaction? Yes, or tryptophan might very well be good, but the whole point, either you put something that disrupts that beta strand, or you put something here that's so large that they don't really want to bind. And that's frequently how people develop drugs. There is a famous drug called Lispro, which is a form of insulin. I'm not sure you know about insulin, but when people are diabetic, one of the problems with human insulin is that it takes quite a while for the insulin to reach its functional form. Insulin is frequently a hexamer in solution or something in the body. This has to dissolve first into dimers and then monomers. And that takes like 30 minutes or an hour or so, which is part of the problem, that it's hard to administer the dose correctly. And I think it was Eli Lilly who had this. Lispro is their internal name. It has some other market name, humane insulin or whatever. They just took two residues, a lysine and a proline, I think it is. That's the reason for the name, and swapped the order of them in a coil. And for whatever reason, this influences the insulin's ability to dimerize. And suddenly it doesn't dimerize as well anymore. And if it doesn't dimerize, it's going to be in the monomer form. And if it's in the monomer form, it acts directly in the body. Super simple. But of course, somebody had to be pretty smart to realize that those are the two residues to do. Let's do it. And at that stage, it's based on modeling. Somebody likely sat down and looked at that. You know what? Let's try to swap those two residues. That's kind of dangerous. Can you make a drug just based on a computer model? So that's, of course, the whole point, right? The second you have the idea that, oh, this would be really smart, then you can tell the lab, guys, have you try the sequences that and see if that works better. And it worked better. And then you're done. And once you are at that sequence, it doesn't really matter how you came up with the idea of swapping them. It just works. It's 10:30. Let's take our 20 minutes and meet here 10 to 11. And then I'm going to continue talking about alpha helices a lot too. I finished the beta sheets before the break, and I'm not going to go back to them, but just to explain, there are lots of reasons why alpha helices are different. We went through a lot of them both the week before the Easter break and the last few days, and you know everything about the properties of the structures. The only thing that we maybe haven't mentioned really that all these hydrogen bonds in the beta sheets leads to lots of constraints because they are non-local, right? So that the more long-range distances that you're freezing and saying this has to be exactly 1 nanometer or whatever it is, the more you restrain different parts of the protein together. While alpha helices, formally they have just as many hydrogen bonds since every residue participates in 2 with the NH group and the CO group. But since all of these are local, it meets the much fewer global constraints. And this is the reason why alpha helical structures, surprisingly, although it's easier to understand how an alpha helix folds, this means that their larger structure is going to be more diverse and more complicated than the beta sheets. So in general, alpha helices, the only requirement we have here is that we somehow need to pack the helices together. There are usually not, at least not a lot of hydrogen bonds between different alpha helices. And here you probably see, right, that any time two helices cross, they appear to cross at an angle that is like 30 degrees or so, that I spoke about before. So this diversity is certainly more important than particularly when you have mixed structures with helices and beta sheets, it's going to get even more complicated. There are some channels there are some structures like hemoglobin that are just seems to be large blobs of helices packed. In particular, membrane proteins that I'm not going to talk about today, that we will look at tomorrow, they frequently have these helices that goes through the membrane that I'm sure that you also in the bioinformatics course. And then we somehow tend to arrange the helices around a pore or something that you want to transport things in. But similarly, we actually have very simple, plain 4 helical bundles or something, just collections of 4 helices in nature. They're not entirely parallel, so they do make a bit of an angle there. You can take those 4 helices in turn and start to subdivide them. Eventually, this is how you classify proteins in nature. So all these are examples of 4 helical bundles. Cytochrome C, TMV coat protein, and hemerythrin. In all these cases, adjacent helices are antiparallel. So you go up, down, up, down, up, down. That's always the case for these 4 helical bundles. I'll just show you briefly. So which one of these do you think is most stable, or which one, is there any of these that you find that the structure is a bit surprising, that it looks strange or something? Yeah, that looks like it should be really floppy or not particularly good, right? There is a reason for everything in nature. This almost looks like a wedge. That's if it's thin here and wide over there. The first one is on the left, cytochrome. Cytochrome fold is a gigantic fold of proteins, and it's usually related to electron transports or binding methods. This is a very fun project that we worked on some 15 years ago at Stanford. We were actually supported by DARPA, which is the US Defense Research Agency. And we were trying to do fold recognition at the time of the entire organism, so basically trying to determine every single fold in Shevanella onedensis MR1. Why on earth would the US Defense be interested in something like that?
Biological weapon.
Well, let's see. This one can bind to metals, and it's likely a bacterium that can chew radioactivity. And this bacterium has more cytochrome folds than any other known organisms. We still don't know, because then the Iraq War happened and they needed the money for cruise missiles instead. So I I've been able to waste some research funding that was spent on ours instead of cruise missiles, and that gives me a bit of a good conscience. So we never got further in that project. That's the danger with DARPA. If they feel that they need the money for something else, they will just take it back instantly. But the point is that there are lots of really complicated processes in nature that we have no idea how they work. It's still one of them. We have no idea how bacterium interacts with radioactivity or heavy metals. And it's kind of important because heavy metals is a common source of pollution, right? Even in drinking water. So what if you could get bacteria to somehow absorb the heavy metals? Because once the heavy metal is in bacteria, it would be much easier to filter it out of the water. Unsolved problem. Tobacco mosaic virus. Tobacco plant leaves. All the black stuff here is a virus that has attacked the plant. Absolutely horrible for tobacco growers. And you can already in the 1940s and 50s, people were able to identify this virus in an electron microscope. Do you see these long rods? It's all a virus. And you can keep amplifying it. And at the end of the day, this is what the virus looks like. It was Rosalind Franklin who determined. These small proteins, do you see that it's a small wedge? So each and every one of these small viruses is a four-helical bundle. This is frequently how nature does it. So viruses in general, they're coated by something. What is the red stuff in here?
RNA.
Exactly. So RNA, a virus is dead, right? A virus isn't alive, but still it's a dead particle that contains genetic material. I love viruses, but they're kind of exactly halfway between life and death. So there is absolutely no cellular process or anything in life. A virus literally just consists of the genetic material and then sort of coat protein to protect that. But then, of course, depending on what coat proteins you have and everything, these viruses get really good at infecting other cells. And then when it affects other cells, they take that RNA, inserts that into the other cell. What do you think that RNA codes for? Tobacco mosaic virus, right? So that it codes for this protein. So that then you have more RNA and more protein, and then you have more viruses. It's an extremely simple life form that way. Absolutely beautiful, remarkably efficient. But the reason is, of course, if you're going to pack this, you'd better have something wedge-shaped if you're going to pack it in a circle around this, right? So that's why the protein looks the way it does. Had this been a normal four-helical bundle, it wouldn't work. Hemoglobin, we've talked a lot about before. I'm not going to go back to this, but hemoglobin is a very amazing molecule because it's such an insanely good binder of oxygen. And I will come back to that later on why. There's also different organisms have different properties in binding hemoglobin. And there's a related myoglobin too, because where you're a whale, if you're a llama living up in the ants, that hemoglobin binds oxygen stronger. Why? There's less in the air. So that, and again, I have no idea how nature does it. Fetal hemoglobin binds oxygen better than normal human hemoglobin. Why? Well, that's certainly true, but a fetus doesn't breathe. How does the fetus get its oxygen? From the mother. So it had better be able to steal oxygen from the mother's hemoglobin, right? Otherwise, the oxygen would go in the other way, which would not be good. But that's, of course, that would not work if the mother, once this fetus grows up, if this is a female, right? If by the time that person is pregnant again, you have to change this. Because when you're grown up, you need to have weaker hemoglobin or you will never be able to be pregnant and support a fetus yourself. So this is something that has to change. And you have both fetal and normal hemoglobin in your genes. But depending, this gene expression changes during your life cycle. And exactly why it binds oxygen stronger or weaker, I'll come back to later. So this is a single unit, 6 helices that essentially forms a tetrahedron. So it's like a shell around it. And then you have this small protoporphyrin heme group in the middle there. And then the yellow iron. And that's actually an iron. That's an iron ion, really. Myoglobin consists of just a single subunit, while hemoglobin consists of 4 subunits. Why on earth is that? I think I'm going to come back to that in like 2 or 3, 4 lectures from now on. But this, of course, has to do with how you deliver oxygen. Where do you have myoglobin and where do you have hemoglobin?
Myoglobin is in muscles?
Yes, and hemoglobin is where?
In blood, in red blood cells.
Yes, in your blood, right? And the hemoglobin somehow has to be able to deliver the oxygen to the muscles. Like if otherwise hemoglobin is great at binding oxygen, but at some point it has to release it. Otherwise you would just have lots of oxygen dissolved in your blood. So hemoglobin needs to be able to get the oxygen to bind in the lungs, but once you're out in the muscles, you need to release it. And this can't be any complicated biological process. This has to work all by itself. And this is how nature has evolved the molecules. So they have slightly different properties. Exactly why we'll talk about later.
Is it because some hemoglobin is 4 and then it's like this cooperative system, like binding one, someone enables to bind the second?
Exactly, and it's so remarkable. It's a remarkably cool process, and it's just a freak of nature that these hemoglobin and myoglobin just turned out to be the first two proteins that we ever got structures for, and that's a coincidence. It's a remarkable coincidence. But again, it's a very typical way that you create a protein. So where do we create these proteins from? Why do they have the amino acids they have? Yes, and this sequence comes from DNA. And by now you've taken all these courses. You should know what an intron and an exon is, right? Do all organisms have introns and exons? Why not? Yes, you're not as cool as bacteria, sorry. You might think that it's very easy as a human to think that you're the height of evolution and everything. First, you're rather embarrassing compared to a virus, right? That virus doesn't need any coffee to work. A bacterium, Jesus, why on earth are you wasting all this space and everything? Can't you do you seriously need to keep changing the proteins you express during your lifetime? Can't you just get to work and work? No, it's bacteria. I'm actually not joking. Both bacteria and viruses are far more efficient organisms than you, partly because they evolve so much more rapidly. So it's like we are the junkyard of evolution because we evolved so slowly. They're far more beautiful organisms than humans. We do have one or two advantages ourselves, but in different ways. So eukaryotes have these exons and introns, so they have regions that we express genes in, and then introns that are not just junk. Today we know that they are responsible for quite a lot deciding on how much protein we are going to express and when we express them. For instance, this fetal versus adult hemoglobin and everything. So what do you think these blue, green, and red regions correspond to?
Different domains.
And that's such a good answer that is completely wrong. This seems so natural, right? If you have red, blue, yellow, and then I have this helix, the first helix, the second helix, and the third helix in your protein. And here's how we call it that. It has nothing whatsoever to do with the structure. And the reason for that is on the previous slide, that the introns are cut out long before you get to the ribosome and actually start to assemble the protein. So that introns and exons, this is the only slide I ever talk about it. Important for deciding when to express it, they have nothing whatsoever to do with structure. And most importantly, the cut is not here. For hemoglobin, it actually turns out that exon 2, the middle one, the blue one, this part can bind a heme group itself. It doesn't work that efficiently, and in particular, it doesn't bind oxygen. So that's, again, obviously why nature needs all three of them. I already mentioned this helix pair packing a little bit when I spoke about early structures, so I'll skip that slide. And you remember this, I said that you can pack the helices, but you can actually turn this the other way too. So it turns out that you get something that's either roughly 50 degrees between the two helices or roughly 20 degrees between them. And if you think that that's just my hand waving or something, just go to the databases, but you can look this up and it works beautifully. You get exactly these helical crossings. So that almost all helices, they group into very beautiful cases, and it's true for all membrane proteins and everything. So this works great. And in particular, this means if you now have a small helical bundle or something that you want to create, this is essentially your first building block, because this is going to decide how can you pack two helices relative to each other. If you now have two such pairs, how can you pack four helices against each other? So why would it be important to pack four helices?
Is that the simplest way you can make a whole?
Exactly right. So two helices, they can certainly interact with each other, but there is no inside here. They just pack against each other. Even three helices would be complicated because there's still no inside. And sadly, I run out of pens here. But the second you have 4, you're going to start having some hole in the middle where you can put something. And if we're going to do protein design, that's a pretty darn good start. So we take these helices, assuming that you would like to bind something like a heme group. What do you think that protein could do?
Transport iron.
Yes, or if you're not interested in transporting iron, there's something else you could transport after the fact that it's bound in iron. Oxygen. So you could kind of make artificial blood, right? The way you do that, to get that to happen, there's quite a lot of hydrophobic residues, and you're going to need this whole bundle to be stable. So we would like hydrophilic residues on the outside and hydrophobic on the inside. Remember the thing that I told you, that there are these helical wheel structures, right? So if you now, these numbers just tell you how the residues will be placed on a helix, and then we make sure to put lots of leucines on the inside between the 4 helices, and then charge groups on the outside. And once we have those, we just place those out in the residue. We have one cysteine that's bound to the porphyrin group here, and then you sequence that. The porphyrin is of course not in your genome, and then you put this in the bacterium and try to sequence it. Do you think it works? It does. You can create artificial blood. Why would that be useful?
Is blood a bit of a commodity?
It's not just a commodity, right? The blood is a live organism. There are cells in it, which of course means that it can carry infections, and we're pretty good at detecting, say, HIV a couple of months after you've been infected. But if you were infected yesterday, there is no test in the world that can detect that. So if you donate blood, what you do is that we typically try to save that blood a couple of weeks before we give it to somebody to at least see if you had something, but it's not going to catch HIV, for instance. And that's why we have all these rules that might seem horrible, that if you belong to a risk group, we prefer that you do not donate blood. It's not based on discrimination, it's just we can't detect these things until it's too late, and therefore we prefer not to take risks. There are of course also lots of parts in the world where it's not safe for something, or you don't have enough blood donors, or if you have a major disaster. So the whole concept of being able to create blood in any amount you want, that would be pretty neat. Because this blood you won't have any worries about specific antigens or that could be rejected. You could tailor make blood. The problem is that it's not quite as efficient as normal hemoglobin. So in the rich part of the Western world, when we have fairly good ways of controlling blood, so we still prefer to work with human blood. It's still better. But still, that's the key word there. That doesn't mean that you can't create something that's even better in the future. And in principle, we could tailor make hemoglobin or something, right? It's not quite, you will need an entire red blood cell too, so that we're not there yet, but give it another 10 or 20 years, and I think you're going to see way more completely artificial proteins that are biomimicking that way.
One question. So this assumption that an artificial protein couldn't be recognized by the immune system.
Now, I'm not saying that it can't be recognized, but you remember, it's artificial, right? So we can design it. Of course, it will be recognized by the immune system. When it comes to blood, for instance, if you donate blood to me or vice versa, there are lots of factors in this blood. And in particular, there is the main blood group, right? O, A, and B. And A and B are antigens here, and they're common antigens. So that you can't, if you have blood group A, that means that you have antigen A. Let's see, yes. So I have blood group O, which means that my blood is nice in a way. Anybody of you can get my blood group because I don't have any of these antigens. I actually happen to have O and then rhesus negative too, and that's even better. That's even better from the point of other people, because my blood can be donated to anybody. I don't have any antigens. The only problem is that I can't receive any blood except ORH minus, because I would not like AB plus blood or something. So when it comes to blood, you certainly recognize it, but the immune system is not binary, right? So that there are some of these antigens that are so common that we know that you would react to that, and it's not the hemoglobin you would react to. But no, in general, you're right. What do you think an antigen is? Yes, it's a protein. That's how you make vaccines. You take the virus and you pretty much grind it down in a food processor so that it's no longer alive. And then you inject this in horse or something. You get antibodies, and these antibodies you can just have as gamma globulin and inject. or you can try to grow these the vaccine then in eggs or something. Oh, sorry, you grow the virus in eggs so that you get enough of it. So this is all about getting your immune system to recognize the parts of the proteins. This can be used for something else. Small proteins such as alpha helices, if they're hydrophobic on one side and hydrophilic on the other, they can actually work as emulsifiers, protein emulsifiers. So why would that be important? There are plenty of emulsifiers in the industry. Why would it be need to have a protein emulsifier? Yes, you can, and particularly you can eat it, right? Digest it is important, but it's also you don't get sick from it or anything. It might even taste good. So that's frequently what we use in low-calorie products. Like these modern margarine or something with 20% fat. How can you have 20% fat? That would be 20% oil in water. Based on what you know about thermodynamics in this course, it's impossible to dissolve 20% oil in water. But that's because you have some sort of emulsifier that sits between the water and the oil. But that has one drawback. So first, not all emulsifiers are proteins. There are many of them that are small lecithins and fatty acids. But in general, using proteins as emulsifier is great because you can tailor make them any way you want. And then they have one horrible drawback. A horrible drawback. You're not going to die. What happens if you try to fry in low-fat products?
You will break the protein.
Yes, the protein denaturates, right? And that's why it looks beautiful, it spreads great, and then you put it in the pan, and before you know it, you have a mixture of water and fat because you broke the protein. And that's why there's quite, you can imagine there's quite a lot of development in the food industry here. Can you make them more stable so that you can fry, for instance, or at least boil it? Do you know what this is? And I was a bit nasty. I'm going to have some more images, but I didn't print them out because then you could guess. What type of structure is it? Yes, lots of beta sheets. So this is part of an antibody. Antibodies are mostly, actually probably even the whole antibody. Antibodies are mostly beta sheets, and these are then some regions binding antigens or something. Do you know what this is more?
What is what?
Do you know what it is more? Well, you can't see it here, but you can. It actually turns out that there are lots of disulfide bridges. So this is a very complicated multi-domain protein that you use disulfide bridges to stabilize this. And it's not that beautifully drawn here, but it actually corresponds to having this entire domain with this Y-shaped part. And here you have these antigen recognizing regions, and this is the backbone of the antibody. Do you know what this is more? There is a reason why I'm showing you this. It's not just for protein structure. No, well, actually it's a good point, but many vaccines are virtually all vaccines are related to antibodies. Typical vaccines work by injecting a very small part of the destroyed protein or something in your body so that your body develops its own antibodies. So you could say that this could be an antibody derived from blood that corresponded to a vaccine. Gamma globulin or something would rather be the type of inject. But no, that was not what I was thinking about.
Monoclonal antibody?
It is actually a monoclonal antibody. Most antibodies are monoclonal, right, if you look at an individual molecule. But when you express a monoclonal antibody, that means that you express, this is completely pure. It's an antibody that every single antibody in a small flask or something is exactly identical, and they will only bind one specific antigen. It's a bit expensive to develop and everything. And this is actually a drug, the Humira, adalimumab AB. Subcutaneous use only, so it's a small pen with a needle that you inject. This is a medication used for rheumatoid arthritis and psoriatic and a bunch of things, Crohn's disease, ulcerative colitis. So this inhibits part of your immune system, which is called the TNF inhibitors, tumor necrosis factors. How long does that one live? That's the reason I asked you before. So when you inject this, it stays around in your body for maybe 2 weeks or so. And then you need more. So, Yes. How much money do you think people are making from this?
Use it for so many diseases.
$12.5 billion last year. This is the world's most selling biological. Do you know what a biological is? Do you know what the classical drug is? Yes, aspirin, right? 1 or 2 small hydrophobic rings or something. It's hydrophobic because that hydrophobic things tend to bind to other small binding pockets. Most signal signal substances of your body are usually small and hydrophobic. Do you know what the molecular weight of aspirin is? Ballpark. So you frequently measure these molecular weights in atomic units, right? And then Daltons. And aspirin is roughly 180 Daltons or so. Do you know what the molecular weight of the thing that I'm now hiding behind that one is? 150,000 Daltons. This is a completely new type of drug. 20 years ago, nobody developed drugs like this because it was too complicated. Because you need to target Humira in particular. I'm not sure whether this is computationally designed, but there are a bunch of companies in the world now that are trying to design antibodies to specifically target one cancer cell when you know what the antigen looks like. There is no natural antibody for that. But the question is, can you then tailor-make these surfaces, even with computer simulations, so they get this antibody to bind one specific thing stronger than another? And there are drugs like that in the pipeline now. It takes, you remember, pharmaceutical development is slow. So from the point things work in the lab until they are on the market, it's at least 10 years. But this is a revolution happening. So 2 years ago, there are more biologicals being patented than there are small drugs. So virtually all I wouldn't say all, but a vast majority of new drug development is focused on biologicals. So biologicals literally means proteins. That we're making protein drugs instead of small peptide drugs. So everything in sequencing and everything is going to lead to a revolution in this field. 10 years from now, and I think it's going to be very rare that we develop new small drugs. But my point here is that forget about the car industry. It's an insane amount of money. There's just no narrow research topics. There is one really big problem here, though. Can you imagine what that problem is? If you're a pharmaceutical company.
Needs to be injected.
Yes. It's really bad because now you need to get a patient to visit a doctor every 2 weeks. Well, it might be possible to inject. So let's see. How many of you would spontaneously like to take injections every 2 weeks? Yeah. Not that it's expensive. This will possibly work in the Western world and everything. It's not a pill. Pharmaceutical companies love pills. First, because eventually there's a patent, say Prilosec or omeprazole, which is called Losec in Sweden. Astra's blockbuster drug. For several years it was just a pill, right? Now you can get it without a prescription in the pharmacy. It's great. They don't make as much money when the patent protection has expired, but they're selling way more of it. Do you think that people are going to go and buy that in the local supermarket? So this is a problem. Why do you have to inject it?
Because it gets digested.
Yes. And this is another one of these, I touched upon that in the early part. If you could start making these type of chemicals, biological drugs, but put them in a pill, you would make insane amounts of money.
Like what this nanopore, nano, like this encapsulation are doing? Like
Yes, so there are a couple of different strategies. One of them is to somehow try to put this in a delivery mechanism that could be a vesicle or something, lipids or something around it, or some other pore to protect it. Another possibility that we're actually working together with a company, purely on the research side, I don't have any financial interest in it, it's actually to apply patches on your skin. Because that is by far one of the nicest ways to deliver drugs. You get a small continuous dose administered in your blood all the time. The problem with that is that only works for small and relatively hydrophobic molecules today. And why? Well, we have an idea, right? That's because they have to pass through membranes. But the question is, can you design components of drugs and everything to make them pass through membranes more efficiently? Because that type of, you're not going to get antibodies deliver that, right? But it's a very cheap and efficient and a simple way to deliver drugs. Lots of work remains in this area. So that So you can kind of see there are two parts here. One of them is the actual biological function of the drug. The other is kind of drug delivery. And drug delivery, in many cases, there's actually more money in that, because if you find a good drug delivery mechanism, you can use it for hundreds of different compounds. So what you would do if you're a pharmaceutical company, let's assume that you have developed a drug, and so you have this awesome new drug that is super good, but then you would seek out another company specializing on drug delivery, say, hey, we have this particular antibody, could you help us deliver this in an efficient manner? And in this case, they failed, so they have to inject it. And of course, if they have said A and B, we're not going to say gamma, but mix A and B. It's virtually impossible to mix a single beta strand directly with anything, because that one can't form hydrogen bonds. And it turns out that when you start mixing things, is you have either alternating helix and sheet or completely different helix sheet parts. And you should remember those. I'm not sure, either you did it in bioinformatics, so A/B or A plus B. So A/B, these are the type of structures that are sheet, helix, sheet, helix, sheet, helix, sheet, helix, sheet, helix. And the reason why they are that is that these sheets really form one continuous sheet, right? They're virtually always parallel, because you go up with the sheet, down with the helix, up with the sheet, down with the helix, up with the sheet, down with the helix, etc. This is a very classical such structure called Tim barrel that can bind things in there. Alcohol dehydrogenase is another fun molecule. Do you know what this one does?
Processes alcohol.
Yes, it breaks down ethanol in your body. In large parts of the Asian population, they're deficient in it. And that's why, for instance, that in particular Japanese population, you get intoxicated very easily. You can't stand as much alcohol, because you don't really have, like, it's not an important, apart from the Nordic countries, I guess, it's not an important part of your body to be able to drink copious amounts of alcohol. It's kind of a freak of nature where we have it and they don't. But the point is that this is not cultural, it's genetic. There is another cool fold called the Rossman fold, and that's also a mix beta alpha, beta alpha, beta alpha. So you have one beta sheet in the middle and then alpha helices on both sides. And that actually creates more binding pockets on both sides of this sheet. Beta sheets are awesome for binding pockets, and there was Mike Rossman, found it in the early 1970s. Actually, I have a slide on that. It's similar here, right? So that you have one binding pocket here and one binding pocket in the middle. And it's very common to have these binding pockets right next to the sheets. And these places are usually awesome active sites or binding sites. Why are they awesome active sites? Well, that's not easy to guess, but remember all those loops in right next to a beta sheet, right? Those loops gives you some flexibility to create a pocket or something. It's very hard to take an alpha helix. You can't distort the backbone in an alpha helix, it's rigid. You can't really distort the backbone inside a beta sheet either. That's also relatively rigid. But up here we have some freedom to create, well, nature has some freedom to swap out residues to create a good binding site, and it can be either polar or hydrophobic. So that it doesn't mean that you always see them there, but if you saw a structure like this and had to guess where the binding site was, guess at the edge of the beta sheets. That's usually where you see them. And that's more important than you think, because what you see, when you first see a structure, you're going to see a structure like that. There is no, you might be surprised, but there are no blue arrows in the protein data bank telling you where things bind. People determine a structure, and then you need to start guessing. Where might things bind? Because you might know that it's an important receptor. How do you test that? You can simulate it, but I was trying to test it. You can actually test it for real to see what you can even simulate it, but assuming that you find a simulation and you find a residue here that is your candidate. Is that right?
You can mutate it out. And then
Yes. So the point, if this is an alanine, mutate to say a tryptophan or something, change it completely, right? And so if you change the residues around that site and absolutely nothing happens, your prediction was probably incorrect. But if you start mutating the residues and that completely changes the binding, you were likely right. So we've said A/B, we can say A plus B too. The only difference here is that here you have one part of the protein that's beta strands and another part of the protein that is alpha helical typically. So the point, never alpha, beta, alpha, beta, alpha, beta. So you have at least 2 or so of them. But by far the most common is like you have here, that you have sheet, 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 and then 2 helices. And in this case, they usually fold independently of each other. Remember what I said about the phase transitions? Nature prefers this because the sheets stay together. It's actually quite okay to have the other ones too, but even there, you never mix one strand directly with an helix. The sheets want to stay together at any cost. These are very common when it comes to DNA binding proteins. So-called TATA binding proteins. So TATA is this motif in the DNA, right? So zinc fingers and everything. So these proteins, they love to bind in these grooves in the DNA. I'm not really going to talk too much about that. But these are important because they can initiate transcription. What could you use that for? Well, assuming that you have a gene that for some reason it's expressed too much, right? Or a gene that's bad or something. You should be able to, if you can find something that binds to the right sequence here, you should be able to silence that gene. Because if you bind something else, you can't initiate transcription at that point. And that's also what people try to do frequently in cancer or something, that if there is a known mutation that's bad, can you silence that mutation and by trying to bind directly to DNA? It doesn't really work too well, to tell the truth. So these haven't been exceptionally efficient as cancer drugs. But the whole point, what all this comes back to, that if you want to start interacting with the cell, you'd better find the process that goes on and find a protein that can bind to it. Every single interaction in the cell has to do with protein binding at some point. In many cases, we don't know what the structure is, but that doesn't change the fact that it is protein binding.
Why didn't it work so well? This has to go inside of a nucleus, or is it
Possibly. So that cancer is a super complicated disease. And I think the mistake that we've all made, I haven't worked on cancer myself, so I don't really know the details of it. What you should be aware of is that there are so many different factors in cancer, things protecting DNA, things splitting DNA, other parts of the cell machinery that goes wrong, so that this whole hope of being able to find 1 or 2 miracle drugs that cures all cancer is not going to happen. So it's a collective process that involves the entire cell's natural life cycle, right? How a cell divides. And the problem with cells is that, again, nature has designed itself to be robust. If something happens to bind to your DNA, the cell should not immediately die. Nature is kind of like an airplane, right? That they always try to have a second route or something, that it should be able to survive no matter what it does. So that cells are awesome at surviving. And the problem when it comes to cancer is that you're not going to try to fight the nature at something that the entire cell has been designed for, to survive. And that's why it's virtually impossible to find one single process and fight just that one. So last year's Nobel Prize in Chemistry was related to this, right? Thomas got the prize for what they actually discovered, that DNA naturally breaks down, and then there are these repair proteins that repair DNA. And that was kind of amazing. But we all thought that DNA was stable for billions of years, but even DNA spontaneously breaks down. So how do you think you could use that to form a drug?
You can target the repair to somewhere after something you don't like?
Yeah, something like that. So the problem with what people ended up doing, it's very easy to go wrong here too, right? Because you think that, oh, should you try to create the protein? What actually turns out, you pretty much do the opposite. You knock out the repair proteins. That sounds horrible.
Only in the cancer.
No, you knock them out in all cells. You can't. It would be great if you could do it only in the cancer cells. That doesn't work. So didn't you kill the person now?
But most cells don't divide as rapidly.
Exactly. So the point is that cancer cells divide a factor 1,000 more rapidly. So for every normal cell you kill, you're going to kill at least 1,000 cancer cells. So the problem is, because cancer cells divide so much more, they're going to be much more fragile to anything that hits this procedure. But that's also why I say it's so easy to go wrong. You need to think of the entire cell's perspective. But there are lots of proteins being decided now to try to target different parts of a cell's natural cycle. I think that, and it's also cancer. When you see cancer as a non-MD from the outside, it's very easy to think that we are still fighting cancer, but it's very easy to think that we haven't made any progress. There are huge areas of cancer where we made tremendous progress. In particular, for instance, child leukemia. Just 30, 40 years ago, the mortality in leukemia was like 95% among children. If a 5-year-old got leukemia, they were dead within 2 years. Today it's pretty much the opposite, 75 to 90% survivability because of modern drugs. And which again, it's just 20, 30 years, right? It's an amazing development. I had that part of the toxins. We spoke a little bit about toxins before. Toxins are special, and on one hand they should be super stable, and then they should just bind and somehow destroy another proteins the way another protein work. The point is that many of these proteins, they might look absolutely horrible. The reason why this looks horrible is because it's going to bind something, and that part has been optimized to bind. So that might bring us to another point. That I've shown you a bunch of folds, and I don't expect you to know all these folds by heart, but as you move up in this ladder of successively higher hierarchies of organizing things, right? At some point you don't even want to think about a helix, but you can think of like a globin-like fold. A globin-like fold is the type of fold you have in a hemoglobin, right? You have this roughly tetrahedral shape with 5-6 helices. And at some point you start to realize that nature reuses these folds. Overall folds, so anything down in a family or something, the blue curve here, they would be evolutionary related, but a fold is literally just a shape. And nature, just because two things share shape, doesn't mean that they have the same genetic origin as you would in bioinformatics. Nature just, and there's surprisingly few folds. In 1992, Cyrus Chotia in Cambridge, he made a famous paper called 1,000 Folds for the Molecular Biologist. And he predicted there are probably only in the ballpark of 1,000 folds in nature. That's not a whole lot. Imagine all the number of sequences we have. And we talked a little bit about before, do all sequences fold proteins? We haven't answered that question yet. But even if they do, these billions of different sequences, they will likely fold into a relatively small number of structures, which is a bit strange. So Mike Levitt, who I worked with several years, he actually found a couple of years ago that this isn't quite true. As beautiful as this was in 1992, we are a bit over 1,000 folds today, maybe at 1,500 or so. But the cool thing is the number of new folds is shrinking. We're discovering fewer and fewer new folds, which likely means that we're started. There are still some white spots on the map, but we have covered most of it. So we pretty much know the entire folding space. I think that's right now it's getting less and less likely every year that we find something we haven't seen before at all. So fold space is way more limited than you might imagine from sequence space. So the last thing I'm going to say that if fold space is so limited, can we design things to fold? Sorry, that is a bad quality image, but if sequence determines structure, determines function, the question is at what point do things change over from one fold to another? Because it's very obvious if I change one residue, nothing happens. In bioinformatics, what do you usually say? At what point can you start to detect that things have, say, the same evolutionary origin? 30% or something. So that you should be able to change at least 30% of the residues, sorry, 2/3 of the residues and still say it's the same, right? But there are examples. Lynn Regan is at Yale for many years. They can keep 50% of the residues identical in a protein, but still turn that into that. So all those things are, again, no rule in biology without an exception, that all residues are not created equal. Some residues are more important than others. And in this case, it was, of course, completely fake in the sense that you decide this in the lab. But in theory, this could happen in nature. Nature might, in theory, end up swapping 50% of the residues in a protein. So occasionally, a change in sequence actually results in a change in structure. It's very rare. And it's one of those things, if you see something you don't understand, don't think that this is the reason, but you should be aware that while things are robust against single changes, at some point you change enough and your way you have changed enough, you will suddenly get the structure change.
Wouldn't that lead to such a drastic function change that it would most likely be selected out?
Yes, sure. Unless there's a small binding site or something in theory, of course it might have, if you're really lucky, you would have the binding residues around it. And again, 1 time in 1,000 or something, you might have a better property there. And then this one would gradually die out instead. Yeah.
We can see that there are like similar binding sites in very different proteins, right?
Yes, and that has to do, for instance, a good example, hemoglobin. If you're going to bind these iron group, there are only two types of residues that are really good at doing that, histidine and cysteine. If you only have those two, the amount of variation you have is not going to be large. You can choose cysteine or histidine, and those are the ones you're going to see. There are a bunch of study questions there too. I'm going to actually leave you 20 minutes early today. I'm not going to go through those, but I will go through them tomorrow. And what I'm going to do tomorrow, we're going to focus almost entirely on alpha helices, but we're not going to talk about alpha helices. I'm going to talk about alpha helices as part of membrane proteins. In particular, the globular protein part is really good to know well then, because the good part is that it's actually going to be a bit less diversity in membrane proteins, but there we're going to talk much more about function. What do membrane proteins do? Why are they created the way they do? Why do we have the channels and transporters and everything do? And that's going to be fairly biological and a bit of neuroscience too. I think that's all I had today. Do you have any questions?